Welcome to cancerisbadembryo.com. This website provides an overview of the oncogerminative theory of cancer development by Dr. Vladimir Vinitsky. Cancer remains a major cause of death in the 21st century. Our inability to cure cancer is largely explained by a lack of understanding of cancer biology and subsequently a lack of productive approaches to the development of effective treatments. The theory provides a comprehensive explanation of cancer biology and of the 6 core properties of malignant tumors, and suggests a number of research strategies that have the potential to revolutionize cancer treatment. The core properties of malignant tumors are cancer stem cell immortality, tumor cell heterogeneity, metastatic spread of cancer stem cells, invasive growth, malignant progression, and selective immune tolerance to cancer cells. Next, we shall examine these properties in more detail. First property, cancer stem cell immortality. One of the core premises of the theory is that the key to understanding the nature of cancer lies in the exploration of potential immortality of cancer stem cells. The theory considers malignant transformation of mortal somatic cells to be the result of their reprogramming into immortal mimic germline cells that are in fact cancer stem cells. The theory uses the term germinalization to describe this process of reprogramming. Germinalization is the only process that occurs within somatic cells during malignant transformation. Similarly to normal germline cells, cancer stem cells realize their potential immortality during the course of their life cycle. The theory uses the term oncogerminative cells to describe cancer stem cells. In vivo immortality of both normal germline cells and oncogerminative cells is realized during the unique natural life cycle of these cells. The left diagram illustrates how germline cells gain potential immortality as they pass through their natural life cycle, resulting in the development of an embryo. The right diagram illustrates how oncogerminative cells, which mimic germline cells, also gain potential immortality as they pass through their life cycle, in this case, resulting in the development of malignant tumors. An oncogerminative cell is potentially immortal since it develops into a new mimic blastula stage embryo, which is in fact a primary malignant tumor that in turn can give rise to potentially endless generations of metastatic tumors. When developing a malignant tumor, an oncogerminative cell follows the same biological principles as a germline cell developing a blastocyst stage embryo. The stages of a germline cell life cycle are zygote, cleavage stage embryo, morula stage embryo, avascular blastocyst stage embryo, implanted blastocyst stage embryo, fetus, and sexually mature body. The stages of a cancer stem cell life cycle are cancer stem cell, mimic cleavage stage embryo, which is in fact a parthenogenetic cancer stem cell, mimic morula stage embryo, which is in fact a tumor germ, mimic avascular blastocyst stage embryo, which is in fact a tumor spheroid, and mimic implanted blastocyst stage embryo, which is in fact a vascularized tumor. Another core premise of the theory is that cancer is a phenomenon of developmental biology, which we have termed as a desperate asexual self-cloning event. In mammals, this phenomenon manifests itself in a desperate attempt of an oncogerminative cell to realize its potential immortality by cloning the host body, which ultimately leads to the development of a malignant tumor. Definition: desperate asexual self-cloning, a phenomenon of developmental biology whereby an oncogerminative cell develops into a new mimic blastula stage embryo, which is in fact a primary malignant tumor. Second property, tumor cell heterogeneity. The theory explains why a malignant tumor which arises from a single cancer stem cell, is composed of a heterogeneous cell population. A pre-implanted blastocyst stage embryo consists of 3 general types of cells: trophoblastic cells of the outer layer, somatic cells, and primordial germ cells of the inner cell mass. The localization of a cell in the blastocyst exclusively determines its direction of differentiation. A malignant tumor, which in fact is a mimic blastocyst stage embryo, also consists of 3 general types of cells: oncogerminative cells that mimic embryonic germline cells, oncotrophoblastic cells that mimic embryonic trophoblast cells, and oncosomatic cells that mimic embryonic somatic cells. This explains heterogeneity of a cell population in malignant tumors. Definition: a malignant tumor, a mimic blastocyst stage embryo with heterogeneous cell population that arises from an immortal oncogerminative cell as it passes through its natural life cycle. Third property: metastatic spread of cancer stem cells. The theory explains the ability of cancer stem cells to spread throughout the host body and develop into metastatic tumors. A phenotypic property of normal germline cells is their ability to aggregate and develop embryonic germinal tissue. Subsequently, at a certain stage of embryo development, this tissue disaggregates into primordial germ cells that migrate to the embryo's gonadal ridges. Cancer stem cells, which mimic germline cells, possess the same basic behavioral property to aggregate and form cancer stem cell compartments within tumors. Subsequently, these cell compartments may disaggregate into cancer stem cells that migrate into the host's target tissues. This migration is known as metastatic spread. Fourth property: invasive growth. The theory explains the cause of invasive behavior of malignant cells and the basic pattern of tumor development. At a certain stage of embryo development, in humans it ranges 6 to 12 days after ovulation, the trophoblast attaches to and invades into the uterine wall, performing implantation of the blastocyst. Tumor spheroid mimics invasive behavior of the blastocyst. Its superficial oncotrophoblastic cells invade into the surrounding tissues, performing implantation of the tumor spheroid with its subsequent neovascularization. The basic pattern of tumor development is as follows. An oncogerminative cell alone is unable to implant, invade, and develop into a primary or metastatic tumor. Just as a fertilized germline cell must first create a blastocyst in order to proceed with the implantation and subsequent development of an embryo, a circulating oncogerminative cell must first create a tumor spheroid in order to proceed with the implantation and subsequent development of a vascularized metastatic tumor. Fifth property: malignant progression. The theory explains the phenomenon of malignant progression. Due to the metastatic selection vehicle, the ratio of oncogerminative, oncotrophoblastic, and oncosomatic cells in metastatic tumors over time may change in favor of oncogerminative cells and oncotrophoblastic cells. The progressive decrease of the fraction of non-malignant oncosomatic cells in the metastatic tumors underlies the malignant progression. Sixth property: selective immune tolerance. The theory explains the basic nature of host selective immune tolerance to post-implantation embryo and cancer. Through evolution, mammals acquired a key mechanism underlying maternal selective immune tolerance to the antigens of an early embryo, which we have termed as phylogenetic immune tolerance. The cause of maternal phylogenetic immune tolerance to an embryo lies in the absence of maternal immunological memory to the antigens of the early embryo. Cancer, which produces embryonic antigens, usurps the phylogenetic immune tolerance mechanism to escape immunological rejection from the host body. Definition: phylogenetic immune tolerance. Natural maternal selective immune tolerance to antigens of an early embryo that occurs due to the absence of maternal immunological memory to these antigens. Research strategies. The theory suggests 6 new theoretical statements and respective research strategies to pursue in the search for effective cancer therapies. Statement: reprogramming of a somatic cell into a mimic germline cell, i.e. germinalization, is the only process that occurs during malignant transformation. First strategy: explore the genetic and proteomic patterns of the reprogramming of a somatic cell into a mimic germline cell and develop gene therapies targeting somatic cells to prevent this process. Statement: an oncogerminative cell, which in fact is a cancer stem cell, is the only cell that possesses a unique property to develop a malignant tumor by passing through its natural life cycle. Second strategy: develop targeted therapies to inhibit oncogerminative cells, the only cells that are able to give rise to malignant tumors. Statement: a circulating cancer stem cell alone is unable to implant, invade, and develop into a metastatic tumor. Just as a fertilized germ cell creates a blastocyst before implantation, a circulating cancer stem cell must first create a multicellular tumor spheroid in order to proceed with the development of a metastatic tumor. Third strategy: develop advanced anti-cancer therapies targeting the development of tumor spheroids and inhibiting their ability to implant and invade host tissues. Statement: a malignant tumor is a mimic embryo. To have cancer is the same as to gestate a bad embryo. Fourth strategy: consider the interaction between a malignant tumor and its host in the context of interaction between a mimic embryo and its host. Evaluate the diagnostic value of new findings in malignant cells and malignant tumors by conducting comparative studies of these findings to embryo cells and embryo tissues. Statement: embryonic development is a potentially malignant event that is controlled by natural maternal defense mechanisms. Fifth strategy: explore natural defense mechanisms associated with embryo development that control invasiveness, immune tolerance, and the phenotypic profile of cells, and use these natural defense mechanisms in development of targeted anti-cancer therapies. Statements: embryos and cancer tumors share similar defense mechanisms that protect themselves against host versus graft immune response. In the case of embryos, these mechanisms are transient and restricted by the term of pregnancy. Unlike embryos, cancer is protected by these mechanisms indefinitely. In order to get rid of cancer, the body needs help to forcibly give birth to the tumor. Sixth strategy: develop advanced vaccines to overcome natural host immune tolerance to embryonic antigens and restore host versus cancer immune response. As Louis Pasteur said, chance favors only the prepared mind. We believe that the oncogerminative theory of cancer development introduces new and exciting avenues to discover revolutionary therapies to help fight and eradicate cancer. We encourage you to explore our website, cancerisbadembryo.com. Familiarize yourself with the full version of the oncogerminative theory of cancer development, review the latest suggested research strategies, and join our virtual institute. Your feedback is greatly appreciated. Thank you, and good luck.